is responsible for the death of 4,877 people in West Africa. This last May of 2014. I have been asked to find the responsible of this tragedy. So come with me to find the responsible, and I'll show you all the information that I have gathered. But before we go into the biochemistry stuff with our Ebola virus, let me give you a little bit of background, some information that you're the only one allowed to see, but that actually can make the difference. So we have that actually in 1976, Ebola virus was first discovered near the Ebola River in Zaire, which now is the Democratic Republic of Congo. The first outbreak infected actually 284 people with a mortality rate of 53%. Unfortunately, 2 months later, a second virus emerged. And it happened that it has actually a high mortality rate of 88%. At this time, infect 318 people. And the last outbreak is from our crime scene, which was in Guinea, located at West Africa, and it happened last March of 2014. As we know, this is the largest and most complex Ebola outbreak. So going a little bit back, we know that our suspect came from the family Phyllobididae. It's from the genus Ebola-like viruses, from the species Ebola syre, and is classified as a viral hemorrhagic fever. Now, what's actually interesting is that all these Phyllobididae family has a negative RNA. So instead of having DNA as we have, or some bacteria have, most of the bacteria, and some other virus, they have RNA. So they come on two, we can say like two different forms. We have RNA positive, and our suspect has negative RNA. And this is going to be important because actually what means that once Ebola virus get into our system, they can actually or RNA polymerase is actually not allowed, or it can actually synthesize any protein from this RNA. So actually, Ebola is actually really smart. So Ebola did something else about this. And let me tell you about it. So are you ready? You're about to learn from information that is completely secret and confidential. From the crime scene, we were able to take some body fluid samples, and by using a transmission electron microscope, we were able to find the morphology of our suspect. It seems that it has a helical morphology, non-segmented. It's more like a filamentous morphology. We were able to measure it, and it measured 920 nanometers length and 80 nanometers diameter. And it seems like this bad guy was actually identified 30 years ago in 1976. It was actually identified to be part of the family Phyllobididae, which is a virus family that came from the order Mononegaridaeans, we can find that with this morphology, which is a filamentous morphology, it fits actually on the family Philobiridae. However, we have 3 different genus, and with that we have different suspects. So what we did actually is that we know that Ebola virus and Marburg virus, they're the only ones that can produce hemorrhagic fever. But looking at the leg of the virus, we could find that it's actually that our suspect must be from the genus Ebola virus. So now we have 5 different suspects, and from this we can work it out. Now that you're familiar with this bat family, let me introduce you how we actually can say or get good approximation of who is a suspect. So we have here that typhus Ebola virus and Western Ebola virus, they actually don't have any daily rate. So they actually don't kill people. So if we go to our table, we can cross out these 2 guys. You can see how the trends go from Bundibugyo, I'm sorry for the pronunciation, Ebola virus, Sundan Ebola virus, and Zaire Ebola virus, which has the most mortality rate. So we have our nice suspect who actually can kill a lot of people, or actually have the potential to kill a lot of people. So, and how actually we can see who of these 3 guys is actually the one that we're looking for? We have, we have this intuition that tells us that probably this is the guy, but we don't actually know about it. So what we're gonna do is we're gonna do some ELISA tests and some PCRs to actually find the molecular biochemistry of our virus. And this is the results that we got. So you can see that actually virus encodes for 7 proteins. So going back to the molecular biology of Ebola virus, we have the Ebola virus encodes for 7 proteins, and we're gonna see how this works. So we already talked that Ebola has actually a negative sense RNA, which means that the only problem actually is that ribosomes can only use positive mRNA. So what actually Ebola did is actually Ebola carries RNA transcriptase. And this RNA transcriptase is going to transform this negative RNA to a positive RNA. This will allow ribosomes to transcribe that viral mRNA, and that's how actually we got the 7 different proteins. But let me explain you a little bit further. So this is the Ebola genomes. We have that encodes for 7 specific proteins, 7 polypeptides. and one of them is actually a glycoprotein. This glycoprotein is the one that will that will help the virus to actually move or to go to for other cells or to invade other host cells. So um, what we have here is something really interesting. So in order for basically the host, uh, let's put an example us, we have immune system obviously. This immune system, if we saw this glycoprotein, which is it's a viral glycoprotein, is actually going to target our immune system. So what actually Ebola does is it transforms as soon as it came out from the ribosome synthesis, the polypeptide synthesis, it came up as a small glycoprotein, and that's actually going to prevent host antibodies to detect. And since it's kind of a neutral molecule, so it prevent the immune system to actually to target that molecule. After mRNA it did it, we're gonna have the glycoprotein. And so we have that L, the gene L will encode for polymerase L. BP30 will go for transcript. It's a transcription factor. And polymerase cofactor would come from BP35. So let me show you actually what does this mean. So if we look at the structure, because like what the virus wants to do is actually make multiple virus from just one virus. So and it's using the host cell to actually do that. So we got the polymerase L, which is this. That polymerase L, which is the leader region, is the one that is gonna bind to the to the caspid of the Ebola. So it will actually transform to the let's say that a membrane that will protect the virus. Then we have BP40, which is this part, which will give the structure of the virus. And basically polymerase cofactors, transcription factors, that's gonna help the Ebola virus to actually transform or to actually start transcription. Because remember that we have to have a transcription factor to start transcription. So we can tell the ribosome, hey, I want to transcript this. Okay, so the glycoprotein, you see how it is this? So this is actually what's gonna target the host cell. So that's actually the molecular biology. Talking about its ecological role, we get that Ebola is actually a zoonotic disease, which means that normally exists on animals, but can potentially be transmitted to humans. Now there is scientific actually suspect that fruit bats are the natural preservers of the Ebola virus, but it hasn't been proved yet. But like we can see on the life cycle of the Ebola virus, actually starts on the fruit bat, which is the reservoir host for Ebola. Then it can be transmitted to other animals like apes, monkeys, antelope, humans, and because of people can actually hunt these animals and eat their and by eating animals meat they actually get infected. So then human through human, it can be an infection human to human because of blood contact or body fluids. So once a first human is infected, the other humans actually have the potential to be infected through, again, body fluids or even the blood. So then they have two options. Uh, One is dead, and the other one is the Ebola survivors. Now, with the Ebola survivors, they actually they don't got they don't have a healthy life after infection because they actually have some effects like muscle age and stigma and yeah. Uh, well, and now again, going back to the life cycle, we see that Ebola can actually go back and when it infects an animal or a human, it actually start doing the same cycle, transforming its RNA from negative to positive, producing its 7 proteins, and then inviting another cell. So, and why should I care? It's actually really important because obviously, if you get infected, you have the potential to die. But let's see how some of the symptoms. So people that get infected, they tend to have fever, fatigue, muscle pain, hemorrhagic bleeding, and obviously then later on leads to death. So there is only way to be infected, as today we know, and it's from unprotected contact with body fluids, which means anything like blood, um, swor. Now, when I say hemorrhagic fever, that means that actually there's bleeding of our bodies, actually bleeding from mouth, eyes, ears, that which actually results seizures and kidney failure. So that's why it's actually really important to know. So now that we know our suspect, please let me find it out. Let help me find this suspect, so this bad guy can be responsible of what he's been doing. So thank you so much for just getting to this. This was a nice, I hope it was nice information. And remember that this information is still secret information. So thank you so much for helping me out. And I hope you have enjoyed this video. Thank you.